Hello guys, welcome back to another video. In this video, we are going to talk about osteichthys or bony fishes. Kingdom Animalia, phylum Chordata, subphylum Vertebrata, and class Osteichthys. In bony fishes, cold-blooded animals, fishes are cold-blooded, marine and freshwater fishes. Body streamlined, endoskeleton bony, mouth is present, terminal, 4 pairs of gill covered by operculum on each side, skin covered by cycloid or ctenoid scale, 2-chambered heart is present, sexes separate, external fertilization, oviparous and direct development. This is the largest class of vertebrate. There are over 29,000 species of bony fish found in freshwater and marine environment around the world. Bony fishes differ from the fish like shark and rays in the Chondrichthyes class. Instead of cartilage, bony fishes have bones. Bony fishes also have a swim bladder. The swim bladder is a gas-filled sac that helps keep bony fish buoyant. They have paired pectoral and pelvic fins, and all but a few species have bones in their fins. They also have dorsal, anal, and caudal fins. Bony fishes also have an operculum. The operculum is a bony flap of skin over their gills that protects the gills. It opens and closes to help bony fishes breathe when they are not swimming. Bony fishes have scales, and most species have a fusiform body design. That means their bodies are rounded and tapered at both ends. This makes it easier to move through the water. Bony fishes have a lateral line that runs across the side of their body. The lateral line is a series of sensory organs called neuromasts that help bony fishes sense vibration and water pressure and help fish navigate and locate prey. Bony skeleton stronger and more rigid than cartilage, made up of collagen and calcium. The operculum, bony plates that protect the gills. Typically, gill slits cannot be seen. Mouth is present, terminal in position. Class Osteichthyes includes all bony fishes. Like all fishes, osteichthyes are cold-blooded vertebrates. Bony fishes share several distinguishing features: a skeleton of bone, scales, paired fins, 1 pair of gill opening, jaws, and paired nostrils. Osteichthyes includes the largest number of living species of all scientific classes of vertebrates, more than 29,000 species. Osteichthyes account for about 96% of all fish species. Living osteichthyes are divided into 3 subclasses: Dipnoi, Crossopterygi, and Actinopterygi. The subclass Dipnoi, also known as lungfishes, is characterized by an upper jaw fused to the brain case, fused teeth, and the presence of an air-breathing organ that opens to the esophagus. The lungfish's caudal fin is continuous with its dorsal and anal fins. Its pelvic and pectoral fins are long and tubular. The subclass Crossopterygi, coelacanths, is characterized by a type of primitive scales, two dorsal fins and fleshy paired fins that contain skeletal elements. Scientists used to think that this entire subclass of fishes was extinct. Then in 1938, a living coelacanth, Latimeria chalumni, was discovered off the coast of Southeast Africa. The subclass Actinopterygi includes all other living bony fishes. Actinopterygian are characterized by fins that are supported by bony elements called rays. More than 29,000 species of bony fishes have been documented. It's likely that many more, including some deep sea species, have yet to be identified. Primitive fishes that back to the Cambrian period about 550 million years ago. These jawless fishes lived relatively unchanged over the following 100 million years. The Devonian period, about 360 to 400 million years ago, is known as the Age of Fishes because of the abundance and diversity of fishes that appeared during this period. In the Devonian, fishes began to develop jaws and paired fins. All four living classes of fishes and the three subclasses of osteichthys were established by the mid-Devonian. Many species of fish that lived during the Devonian are now extinct. Bony fishes continued to evolve after the Devonian period. Today, the actinopterygians are the dominant vertebrates in the ocean and in freshwater systems. The most recently evolved order of bony fishes include the Pleuronectiformes, that is flatfishes, and Tetradontiformes, that is triggerfishes, pufferfishes, and mollusks. The osteichthyes include the ray fin fishes subclass Actinopterygi and lobe fin fishes subclass Sarcopterygi. Some taxonomic schemes do not consider osteichthyes a formal taxonomic group as it is not monophyletic. Nelson 2006. Although the term is retained for vernacular use for those actinopterygians and sarcopterygians conventionally termed fishes. Most bony fishes belongs to the Actinopterygi. There are only 8 living species of lobe fin fishes, including the lungfishes, 6 extant species, and coelacanth, 2 extant species. Some species of lobe fin fishes have jointed bones. Actinopterygi or ray fin fishes include the most familiar fish such as sturgeon, gar, eels, carp, herring, catfishes, goldfishes, oarfishes, seahorses, cichlids, pickerel, salmon, and trout. The actinopterygi are generally classified into two groups, the chondrostei and the neopterygi, the latter of which includes the teleostei, modern bony fishes, a classification into which most fish fit today. Rayfin fishes occur in many variant forms. Rayfin fishes have many different types of scales, but all teleos, the most advanced actinopterygians, have ellipsoid scales. The outer part of these scales fan out with bony ridges, while the inner part is crossed with fibrous connective tissue. Leptoid scales are thinner and more transparent than other types of scales and lack the hardened enamel or dentine-like layers found in the scales of many other fishes. Unlike ganoid scales, which are found in non-teleose actinopterygians, new scales are added in concentric layers as the fish grows. Sarcopterygi are traditionally divided into the lungfishes having elasmoid scales and coelacanth having cosmoid scales. The Sarcopterygi are the low-fin fishes and the tetrapods. The name Sarcopterygi or sometimes Crossopterygi used to refer only to the fish member of the group, but since it has become clear that the tetrapod evolved from Sarcopterygian fishes. The precise relationship among all the Sarcopterygi are still a matter of debate, and there are many competing hypotheses. The earliest low-fin fishes are found as fossil in the Lower Devonian, and by the end of this period all major lineages had appeared. These early low fins were fast swimmers with a heterocircle tail, meaning that the tail fin was asymmetrical and larger on the dorsal side. In the living low fin fishes, lungfishes and coelacanths, the tail is now symmetric, though the fossil record shows that this change happened independently in the two groups. One of the most important characteristics of lobe-fin fishes is the lobe in their fins. Unlike other fishes, Sarcopterygians fish has a central appendages in their fins containing many bones and muscles. The fins are very flexible and potentially useful for supporting the body on land, as in lungfishes and tetrapods. Additionally, the Sarcopterygi is known for having enamel on the teeth. Osteichthys are characterized by a relatively stable pattern of cranial bones, rooted teeth, and medial insertion of mandibular muscles in lower jaw. The head and pectoral girdles are covered with large dermal bones. The eyeball is supported by sclerotic rings for small bones, but this characteristic is missing or modified in many modern species. The labyrinth in the inner ear contains large otoliths. The brain case or neurocranium is frequently divided into anterior and posterior section divided by fissure. Osteichthyes have a lung or swing bladder. They do not have fin spines but instead support the fin with lapidotracheal bone fin rays. They also have an operculum which helps them breathe without having to swim. The fin limbs of Sarcopterygian show such a strong similarity to the expected ancestral form of tetrapod limbs that they have been universally considered the direct ancestors of tetrapods, that is, four-legged vertebrate in the scientific literature. Now, circulation and gas exchange. In case of bony fishes, four pair of gills covered by operculum. Gill arch support Gill filament help in gas exchange, and gill raker trap food helpful during filter feeding. Fish have two-chambered heart. Blood picks up oxygen in the gills and travels to the body through arteries. Blood enters and exits heart through a vein. Counter current exchange system maintains concentration gradient between blood and water. Blood passes through gills in the opposite direction of passing water. Sharks use ram ventilation, open their mouth and swim so they can never stop swimming. Now, buoyancy. Members of class osteichthys passes a swim bladder that is able to fill with air and alter the buoyancy of the fish. The bladder fills by gas exchanged with blood vessels lining the sac. As oxygen diffuses in the sac, the fish becomes more buoyant and rises. The process works the same in reverse. Now, nervous and sensory functions. Like most of vertebrates, the nervous system includes a brain and spinal cord. Sensory structures are widely distributed over the body in addition to the structure for touch, water temperature, vision, hearing, equilibrium, balance, detecting water movement. Fish have external nares. These are openings that lead to olfactory receptors. The eyes of a fish are similar to other vertebrates but have no eyelids. Just like other vertebrates, the receptors for equilibrium, balance, and hearing are in the inner ear of a fish. Lateral line system, sensory pits in epidermis, detect water current from predators or low frequency sounds. Electroreceptions, detection of electric field that the fish or another organism generate, highly developed in the ray and shark fishes. Now, electric ray current that circulate from electric organ in the fish tail to electroreceptors near the head. Some fishes can be electroreceptive but are not electrogenic. Members of the order Apods, eels are elongate, slender fishes without pelvic fin, and in some species lacking pectoral or median fins called electric eel. Due to streamlined shape or streamlined body and mucoid secretion that lubricate its body surface, reduces friction between the water and the fish. Water buoyancy properties also contribute to the efficiency of the fish's movement. Fish move through the water using their fins and body wall to push against the incompressible water. Now, reproduction. Fishes have sexes separate, external fertilization, oviparous, and direct development. Majority of bony fishes utilize external fertilization. Female lays eggs and male drops sperm over eggs. Fertilization takes place and egg eventually hatch. Other fish release fertilized egg that may float, sink, stick to plant matter, or brood inside the parent gills or mouth. Most bony fishes spawn once a year until death. Salmon only spawn once of the age of 5 and later die. Diadromous fishes require marine and fresh water to complete their life cycle. Anadromous migrate from marine to freshwater to spawn. Catadromous fishes migrate from freshwater to marine water to spawn. Now, osmoregulation. Ability to maintain internal balance of salt and water. Higher concentration of salt than environment in freshwater fishes. In freshwater fishes, higher concentration of salt than environment. Highly efficient kidney, fish reabsorb salt from urine. In case of marine fishes, higher concentration of salt outside the body. Hence, gill removes salt from water fish drink. Osmoregulators actively control salt concentration despite the salt concentration in the environment. An example in freshwater fish. Some marine fish like sharks have adopted a different efficient mechanism to conserve water, that is osmoregulation. They retain urea in their blood in relatively higher concentration. So students, hope that enjoy the lecture. Thanks for watching.